Hello everyone, welcome to another learning class of biology. We have already done with transformation experiment that was initiated by Griffith, and finally the conclusion was given by Avery, MacLeod and McCarty. Now we are heading towards the next experiment in the field of genetic material. We will see how this experiment will be done, but the name of the scientist, it is important. The names are Hershey and Chase. Okay, so he was the one and this is the another one. We will see how they proceeded, right? So first of all, they used bacteriophage for their experiment, and just to depict how bacteriophage acts, I have drawn few diagrams for you. Bacteriophages are the viruses that attack the bacteria only. Bacteriophages, they have their genetic material, they puncture their genetic material in the bacteria, and then the genetic material of the bacteriophage gets replicated along with the bacterial genetic material. So you can see in step number 1, bacteriophage is attached to the bacterial surface because bacteriophage is a virus only. It will be attacking only the bacteria. In the step number 2, you can see that the genetic material from the bacteriophage has ruptured the bacterial wall and it is penetrating inside. Now see here at the third step, the genetic material of the bacteriophage, that is shown by blue color, has reached the bacterial inner part. Okay, and then the next step shows the merging, the unison of the bacteriophage genetic material along with the bacterial genetic material. And now in step number 5, you can notice that replication of DNA has taken place, and this replication of the bacterial genetic material has also caused the replication of the bacteriophage genetic material. Right? This process in microbiology you will study in senior classes. This process is known as transduction. Right? Now with the same aim, the Hershey and Chase experiment, it is also known as transduction. So if you will get a question on transduction, please do remember that you have to mention Hershey and Chase experiment. Now as I said in the transformation experiment, that the names of the discoverers, they are very important to learn, because board people, they are giving questions with the name of the discoverers, and the year is worth mentioning. It is very important. So Hershey and Chase, they started their experiment in which year? Yes, it is 1952. Which bacteriophage they used? They use the bacteriophage T2, right? And they use, of course, bacteria for this purpose. Okay, so these are our requirements to do the experiment. Bacteriophage will be there, bacteria will be there, radioactive isotopes, they will also be there. Right, we will discuss why they use radioactive isotopes. So 2 radioactive isotopes were used, one with the 32 mass number phosphorus P32 and another was sulfur with the mass number 35. Okay, so all these are the requirements: bacteriophage T2, bacteria, radioactive isotopes P32 and S35. Another important thing which we should know, which technique they used. So they used the process called centrifugation. Right? Centrifugation is a process wherein the components, they get separated because of their density. So the most denser would be at the base and the less denser would be above, right? Same way we go for this process, and it is done in an instrument which is known as centrifuge. This centrifuge has various columns wherein the centrifuge tubes are there. Okay, so whenever we want to separate any material basing upon its density, we pour that material in the centrifuge tubes. They are arranged in the centrifuge, and then the power is turned on and we set the revolutions per second. Okay, so RPS means revolutions per second. We set it with the help of a button provided on the centrifuge, and then we turn it on. Now over a period of time, we set the time also for how much it has to be run. Then the components in the centrifuge tube, they settle according to their density. So this concept was used by Hershey and Chase. This is the name of the process. This is the name of the instrument, and this instrument used centrifuge tubes. Okay, and we have already discussed about bacteriophage T2, bacteria, radioactive isotopes P32 and S35. This was our basic requirement. Let's get proceeded. Now we need to know the structure of bacteriophage. Bacteriophage, they contain an outer layer, and this outer layer is known as the capsid, right? So this outer part is known as capsid, and this is proteinous in nature. When I say proteinous in nature, means it is made up of proteins, and you know that proteins are the polymers and amino acid, they are the monomers, right? So capsid is a layer and it is made up of n number of capsomeres. So these round structures I am drawing right now, these are actually the monomers called capsomere. So you can see that many capsomeres, they are assembling together, and what are they forming? They are forming capsid, which is a proteinous layer. Okay, so another important thing, the genetic material is confined on the inner side, right? So you know that genetic material is actually a polynucleotidic structure, and this polynucleotidic structure means the polymer, it would be containing monomer as the nucleotide, and you know that one nucleotide is made up of base, sugar and the phosphate, right? So what Hershey and Chase did, they use P32. This P32 they used, and it could be incorporated in the genetic material, right? Because genetic material utilizes phosphate, whereas S35 or sulfur 35 could be incorporated into the capsomere, because these capsomeres are the amino acids, and amino acids, you know, there are few which do contain sulfur in them. You have read about sulfur containing amino acids in grade 11. We read the name of cysteine and methionine quite prominently, right? So S35 could be accommodated in the amino acids, whereas P32 can be incorporated in the phosphate group of the genetic material. Okay, now what they did, they used the radioactive isotopes, and they wanted to check which part of the bacteriophage it entered in the bacteria, and that propagated further. So what they did, they wanted to check whether it was capsid or was it the genetic material that transferred from bacteriophage to the bacteria and then further carried on for the generations. So what Hershey and Chase did, they took different types of bacteriophages T2, and what they did, they made first the outer layer radioactive. So just to show the radioactive nature, I am drawing it with green, and in this they made the capsid radioactive. When capsid was made radioactive, of course, which radioactive isotope they would have used? Please answer in the comment box. I would love to check it. Okay, they used sulfur 35 to make the capsid like this. Okay, so radioactive S35 was used and this made the capsid radioactive. Genetic material in this was like this. So they use such bacteriophage and they left it in the bacterial culture. Bacterial culture means when we artificially grow the bacteria in some nutrient medium, then we say it that we are doing the culturing of the bacteria. So this type of bacteriophage, it was left in the culture number 1. You know that bacteriophage, they have certain generation time, and after that generation time they used it for the centrifugation process. But simultaneously they did another experiment as well because they wanted to come to a conclusion. So in another one, what they did, they used the bacteriophage T2 only, but in this what they did, they made the genetic material as to be the radioactive, right? Because they use P32 for this purpose. Similarly, they allowed this bacteriophage to attack the bacterial culture, and over a period of time the components were used for the centrifugation process. Now when they took the bacteria and they centrifuged it, what they found, of course the bacteria, they are going to be heavy, so they will be settling at the bottom. So all the remnants or the remains of the bacteria, they were there at the bottom because they were much denser. And I have already explained you that while we are doing centrifugation, the components, the materials, they get separated basing upon their density. So what they noted, that the entire radioactivity was associated with the above layer. Now in centrifugation, one more thing I just wanted to tell, the lighter things, they always present at the top, and this part of the centrifuge product, it is known as supernatant, and the part which settles at the bottom, it is known as pellet. So what they found when they used the step number 1, wherein they made the capsid radioactive, so what they found, that entire radioactivity was associated with the supernatant portion. That means they concluded it was not the capsid that entered in the bacteria, otherwise it would have shown radioactivity, the bacteria would have shown the radioactivity, but bacteria were not showing radioactivity. So it proved that capsid from the bacteriophage, it never passed the bacterial genetic material. Okay, now let's see what they concluded in the another experiment. So they grew the bacteria with the bacteriophage, and then they took the bacteria, they did the centrifugation process as they did with the step number 1. Now in this, of course, bacteria will be denser, so it would be settling at the base. And in this, what they found, that radioactivity was associated with the pellet. You know that pellet is the portion which settles because it is much denser. You can see that I have shown green dots, and these green dots means the radioactive P32 was associated. And where they use the P32, it was used in the genetic material of the bacteriophage. So this Hershey and Chase experiment, this was the one that proved that it is the genetic material that is passed from one generation to another. Hope that was useful to you. Stay tuned for one more experiment that will prove the semi-conservative replication of DNA, and this was done by Messelson and Stahl. So in this chapter, these 3 experiments which we are talking about, 1 transformation, 2nd transduction, and 3rd the experiment that proved that DNA replication is semi-conservative, they are worth preparing. They are very, very important. I hope my videos are helping to you. If you have not subscribed to my channel, I would really make a kind request to please do it and share with maximum friends so that everyone should get the benefit for free. Stay tuned for more videos. Thank you.